Welcome to the PubMed Daily Review for May 8th, 2026. Today we scanned 8,226 new open access papers. Here are the top 5 selected for detailed analysis. Trend 1 of 5, multi-domain characterization and modeling. This trend integrates diverse data and methods to comprehensively characterize and model complex systems across multiple domains. This cluster covers 1072 papers. Representative work includes What a Mesh, a call for better science and regulatory oversight, data-driven characterization of knee structures using non-negative matrix factorization of 3D multi-echo UT MRI, and how membranes shape up for lipid transfer. Trend 2 of 5, clinical research and patient management. This trend encompasses clinical studies evaluating patient outcomes, treatment effectiveness, disease characteristics, and quality of life across diverse medical fields. This cluster covers 1810 papers. Representative work includes Do white noise or music relieve pain caused by botulinum toxin injections? Prevalence of oral lichen planus in patients referred to the Department of Oral Diseases and Periodontology in Ljubljana, Slovenia, and driving HCV elimination through dedicated hospital-based teams and pathways. A retrospective and prospective study. Trend 3 of 5, women's and public health issues. This trend investigates health issues, interventions, and determinants impacting women and broader population well-being. This cluster covers 1,700 papers. Representative work includes interventions for preventing unintended pregnancies among adolescents. Prolonged oscillating preoptic area Kisspeptin neuron activity underlies the preovulatory luteinizing hormone surge in mice. And lupus, a leading cause of years of potential life lost in young women. Implications for public health priorities and research funding. Trend 4 of 5, fundamental cellular and microbial processes. These papers collectively investigate fundamental mechanisms governing cellular life and microbial interactions. This cluster covers 1624 papers. Representative work includes geographic clustering and population structures of Campylobacter jejuni and Campylobacter coli in South and Southeast Asian poultry systems. Microbe profile. Durasdinium trincii, a thermotolerant coral symbiont, and evidence supporting the first secondary chromosome in actinobacteria as a hallmark of the Emblea genus. Trend 5 of 5, stroke management and neurological prognostication. This trend unifies research on stroke treatment, complication management, and neurological outcome prediction across cerebrovascular conditions. This cluster covers 2020 papers. Representative work includes European Stroke Organization Guideline on Stroke-Associated Pneumonia, European Stroke Organization, ESO, European Association of Neurosurgical Societies, ENS, and European Society for Minimally Invasive Neurological Therapy, SMIT, Guideline on Aneurysmal Subarachnoid Hemorrhage, and Prediction of Recurrence in Patients with Non-Acute Atherosclerotic Middle Cerebral Artery Occlusion. A longitudinal study using computed tomography, perfusion, and magnetic resonance imaging. Up next, our first paper out of 5. A synthetic opsin restores vision in patients with severe retinal degeneration by Sumerendra K. Mohanty and Vineet B. Mahajan from Nanoscope Therapeutics Inc. 2777, North Emmons Freeway, Dallas, Texas, 75207, USA. Let's break down its key contributions. Imagine a world where inherited retinal degenerations no longer lead to blindness. These conditions are a major cause of vision loss globally, and in their advanced stages, cell damage makes many current gene therapies ineffective. This paper explores a groundbreaking solution, optogenetics. By making the remaining retinal cells light-sensitive, this technology offers a unique, mutation-independent way to restore vision. This was an investigator-initiated, open-label study involving 4 patients who were blind due to retinitis pigmentosa. Each patient received a single intravitreal injection of a novel gene therapy containing a synthetic opsin designed to make their retinal cells light-sensitive. The researchers then monitored their vision, ability to discriminate shapes, and mobility for a full 52 weeks to assess the therapy's impact. The results were truly exciting. All patients showed improvements in their vision, and importantly, they also demonstrated better shape, discrimination, and mobility, indicating real-world functional gains. Crucially, despite this being a highly synthetic protein, no significant safety issues were reported throughout the 52-week follow-up. Non-invasive imaging also confirmed successful gene expression in the retina, validating the delivery method. This study represents a monumental step forward, marking the first time a gene monotherapy has restored vision in blind patients using a mutation-independent optogenetic approach. While these initial results are incredibly promising, it's important to remember the small sample size and open-label nature of this pilot study. Future research will need to involve larger, controlled trials to fully confirm the efficacy and long-term safety of this innovative therapy. Up next, our second paper out of 5. Adapter-based allogeneic CAR-T cells to overcome antigen escape in solid tumors by Q, Pham, and Kuo VH. Saji from Department of Human Oncology, University of Wisconsin-Madison, Madison, Wisconsin, United States. Let's break down its key contributions. CAR-T cell therapy has revolutionized cancer treatment, especially for blood cancers. However, we're still grappling with significant hurdles. Tumors can escape treatment by changing their antigens. Manufacturing these therapies is complex and time-consuming, and their effectiveness in solid tumors is often limited. This paper explores how we can combine two promising strategies, allogeneic, off-the-shelf CAR-T cells, and adapter-based systems, to tackle these challenges head-on. This paper is a comprehensive review, not an experimental study. It synthesizes existing knowledge on various CAR-T cell platforms, including monospecific, allogeneic, and adapter-based approaches. The authors meticulously analyze current progress, limitations, and the potential for combining these strategies to create more effective and accessible therapies, particularly for solid tumors. It's a deep dive into the current state of the art in CAR-T cell engineering. The review found that while monospecific CAR-T cells have achieved remarkable success, antigen escape remains a major challenge, leading to relapse in a significant number of patients. Allogeneic CAR-T cells address the manufacturing bottleneck, but they too fall prey to antigen escape. The authors propose that adapter-based CAR-T cells, which can target multiple tumor antigens, when combined with allogeneic manufacturing, represent a powerful off-the-shelf solution. This unified approach promises to tackle both antigen escape and the complexities of personalized cell therapy production. This review underscores a critical shift in CAR-T cell therapy, moving towards universal, off-the-shelf products that can be readily available for patients. By integrating allogeneic manufacturing with adapter-based targeting, we could potentially overcome antigen escape, a major cause of treatment failure, and make these life-saving therapies more accessible. However, significant work lies ahead in translating this promising concept into safe and effective clinical treatments, especially for the complex landscape of solid tumors. This unified strategy represents an exciting frontier in cancer immunotherapy. Up next, our third paper out of 5, Clinical Variant Interpretation Comparing Two Saturation Genome Editing-Based Functional Studies for BRCA2 by Ju-Hein Shin and Jong-Won Kim from Department of Laboratory Medicine and Genetics, Samsung Medical Center, Sungkyunkwan University School of Medicine, Seoul, Republic of Korea. Let's break down its key contributions. BRCA1 and BRCA2 genes are vital for assessing hereditary cancer risk. However, a significant number of identified variants are classified as variants of uncertain significance, or VUS, making clinical decisions difficult. Recently, high-throughput methods like saturation genome editing, or SG, have emerged to functionally characterize these variants. But what happens when different SG studies for the same gene produce conflicting results? This paper tackles that very challenge for BRCA2. Researchers retrospectively analyzed data from 526 patients at the Samsung Medical Center, focusing on 88 BRCA2 variants that have been evaluated in two distinct SGE studies, one using human HAP1 cells and another using mouse embryonic stem cells. The core of the study involved comparing the functional results from these two SGE datasets with clinical data, reevaluating variants using established guidelines, and assessing how often classifications changed or errors occurred. This unique approach aimed to see if real-world patient data could help reconcile conflicting functional evidence. The study revealed that a substantial 16.9% of BRCA2 variants showed contradictory functional results between the two SGE studies. When comparing major error rates against clinical classifications, the HAP1SG dataset had a lower rate of 15.4% compared to 23.1% for MESSG, although this difference was not statistically significant. Crucially, for variants with concordant SG results, An impressive 93.1% of variants of uncertain significance were successfully reclassified, highlighting the clinical utility of consistent functional data. This research underscores that when SG studies provide concordant functional evidence, it's incredibly useful for reclassifying BRCA2 variants and reducing the number of EUS. However, if SG results conflict, functional evidence should be interpreted with extreme caution, and the HAP1 SG dataset appears to align more closely with patient-specific data. Moving forward, for those challenging discordant cases, additional segregation analysis and long-term patient follow-up will be critical to achieve definitive variant classifications. Up next, our fourth paper out of 5, Intrathecal Mesenchymal Stem Cells in Progressive Multiple Sclerosis, a randomized, double-blind, placebo-controlled trial, SMART-MS, by Christopher Elman-Kvistad and Lars Boe from Department of Neurology, Haukeland University Hospital, Bergen, Norway. Let's break down its key contributions. Progressive multiple sclerosis, or MS, remains a challenging condition with a significant unmet need for treatments that can actually repair nerve damage. Mesenchymal stem cells, or MSCs, have shown exciting potential in lab studies and early trials, suggesting they might help regenerate damaged tissues. This study aimed to rigorously test if delivering these cells directly into the spinal fluid could offer a neuroregenerative benefit for patients. This was a randomized, double-blind, placebo-controlled crossover trial, a robust design for early-phase studies. We enrolled 18 patients with progressive MS across 4 Norwegian hospitals. Each patient received either a single intrathecal dose of their own MSCs or a placebo, with a crossover at 6 months. The main goal was to see if MSCs could improve nerve signal transmission, measured by evoked potentials, after 6 months. Unfortunately, the primary endpoint showed no significant difference in evoked potential latency between the MSC and placebo groups, with a p-value of 0.668. While there was a temporary reduction in cerebral atrophy at 6 months, this effect wasn't sustained. Importantly, the treatment was associated with several adverse events, including 1 serious event and spinal MRI abnormalities in 7 patients, suggesting an acute inflammatory reaction. This study provides important evidence that a single intrathecal dose of MSCs did not achieve the desired neuroregenerative effect in progressive MS. The observed adverse events, including spinal MRI changes, highlight significant safety concerns that warrant careful consideration. Therefore, future research into intrathecal MSCs for MS must prioritize safety and potentially explore different dosing, cell types, or administration routes with extreme caution. Up next, our 5th paper out of 5, Genotype-Guided Antidepressant Prescribing for Patients with Depression, a randomized clinical trial by Catherine V. Blake and Josh F. Peterson from Center for Pharmacogenomics and Translational Research, Nemours Children's Health, Jacksonville, Florida. Let's break down its key contributions. Depression is a widespread and growing health concern, and while SSRIs are a common treatment, their effectiveness can be inconsistent. We know that individual genetic differences can affect how people metabolize these medications. However, despite this knowledge, there's been a lack of clear evidence on whether using genetic information to guide SSRI prescribing actually leads to better outcomes for patients. This study aimed to address that critical gap. This was a large, pragmatic, randomized clinical trial, enrolling 1,460 adults and children with depression across various clinics in the U.S. The core of the study focused on 692 patients who had an actionable genetic profile for SSRI metabolism. These patients were randomly assigned to either receive SSRI, prescribing guided by their genetic information, following established CPIC guidelines, or to continue with standard usual care. The main goal was to see if genotype guidance improved depression scores after 3 months, with longer-term remission rates also being tracked. Let's look at the numbers. At the 3-month mark, we observed no significant difference in the primary outcome, which was the change in PROMIS depression T-scores between the genotype-guided group and usual care, with a p-value of 0.68. However, a different picture emerged at 6 months. The remission rate for depression was notably higher in the genotype-guided group, reaching 48.3% compared to 39.4% in the usual care group, a statistically significant difference with a p-value of 0.02. This suggests a potential delayed benefit. This study provides important insights. While pharmacogenetic guidance didn't show immediate benefits at 3 months, the significantly higher remission rates at 6 months suggest a potential longer-term clinical advantage. This highlights the complexity of depression treatment and the need to consider extended follow-up periods. Moving forward, it's crucial to conduct more studies to understand the sustained effects and optimal implementation of genotype-guided prescribing in routine clinical practice. That's all for today's PubMed Daily. If you found this helpful, please leave a comment, and subscribe for daily paper summaries. See you tomorrow.